All right, I think we'll get started. Thanks everyone for coming and on Zoom. So it's my great pleasure to introduce Heide Ford today, who's a professor and chair in the Department of Pharmacology and also the associate director of the University of Colorado Cancer Center, to be our Cancer Center speaker today. Heide did her PhD at the University of Rochester in the lab of Sarita Zhang while working on a transcription factor, which I didn't realize, MTS1. She then went to Dana-Farber with Arthur Pardee, where she really focused on cell cycle regulation in cancer. She then started her lab at the University of Colorado and really worked her way up through the ranks. Here she started focusing on 6-1, which I'm sure she'll tell us about today, starting with breast cancer and its role in cell cycle, but then also identifying that 6-1 is amplified in many kinds of cancer, so has moved on and focused on ovarian, rhabdomyosarcoma, et cetera. And that's sort of where we started collaborating. We worked together on a project to understand 6-1 and developmental regulation roles in also in development, but also in rhabdomyosarcoma. So at CU, she's really had a lot of leadership roles, really primarily started out in graduate education, which she was really foundational in some of our umbrella graduate programs. And we co-directed that together before I moved here, which was really fun. She's, of course, takes a lot of other leadership roles as an editor of BCR, chairs study sections, et cetera. And I think one of the most impressive things of her CV is that she's won mentoring awards like many times, which I find most impressive. So thanks for coming, Heide, and we're looking forward to your talk. So join me in welcoming Heide to present Enhancing Natural Killer Cell Function to Combat Breast Cancer Metastasis.
So first, I'd like to thank Kristen and Kaylee and everyone who's invited me here and all the familiar faces I've seen. I've already had a great time meeting with some of you who I haven't seen in a few years, know a lot of people here. It's a great place. So I'm really happy to be here. So I'm actually not going to talk about 6-1 very much today. I'll maybe briefly mention it, but I'm going to talk about something a little bit different, a different direction my lab has gone in. That's really been spearheaded by a postdoctoral fellow in my lab. I am not an immunologist. So if there are some immunologists out there, feel free to ask me questions, but you'll probably know more than I do. I am mostly interested in metastasis and tumor progression, but my postdoc has really pushed me into the world of immunology. So it's been a really fun project, and I have to give her a lot of credit for this. Okay, so I'm just going to give a brief background on breast cancer, which I think many of you are probably familiar with. So 1 in 8 women in the US will develop breast cancer in their lifetime. About 30% of those women will develop metastatic disease. The 5-year survival rates of breast cancer are very good if the cancer is localized or regional. But of course, if you're diagnosed with distant cancer, so it's metastasized, then the prognosis becomes much worse, and particularly if that's triple negative breast cancer. And that's the type of cancer we're going to focus on today. And so immunotherapies have become, obviously, many of you have heard about how immunotherapies have become a major way of treating many different cancers, particularly melanomas. And they've been used and approved in combination with chemo for triple negative breast cancer. The outcomes have gotten a bit better in the last few years, especially with specific combination therapies. But in general, immunotherapy hasn't worked as well in breast cancers as we thought it might, and particularly in triple negative breast cancer, where these cancers are thought to be highly immunogenic. They have many mutations. And so why that's not working better in the context of breast cancer is something that we're really interested in understanding. Okay, so I'm going to go just a little bit of background on the immune system and how it regulates cancer. So I'm sure many of you are familiar with the fact that during tumor progression, when people begin to get tumors, it's thought that there's elimination by a variety of different immune populations that are in the body that can notice that the cancer cells are abnormal and eliminate them. But there comes a point at which the tumors become dormant. This is particularly prevalent in breast cancers, in which they reach this equilibrium with the immune system. And then a point at which the tumors begin to progress, they escape immune surveillance and progress and become more metastatic. And we're interested, very much interested in this latter point, like how do the tumor cells escape the immune surveillance? And so this postdoc in my lab, Shira Rosenbaum, this is a review by Rosenberg, not the postdoc Rosenbaum. was very interested in looking at some of these cytotoxic lymphocytes and how they play a role in anti-tumor immune responses. And as you know, a lot of groups have focused quite a bit on CD8 positive T cells and their response to the tumor, but she was particularly interested in these other cytotoxic lymphocytes that are part of the innate immune system, the natural killer cells. And part of the reason for that is that there had been a number of studies that show that the natural killer cells are actually the first responders to eliminate metastatic cells when they hit the distant site. So this is a study performed by another group in which they showed that when you put tumor cells into the tail vein, natural killer cells are the most abundant in the metastatic site. And in fact, if you then deplete natural killer cells from the tumors or from the mice with this antibody ASGM1, it doesn't really do much to the primary tumor, but it dramatically affects metastasis, in that when those cells are depleted, you have much more metastases occurring at the metastatic site. And so what I'm going to talk about today are sort of two different projects, both really spearheaded by this postdoc in my lab, Shira Rosenbaum, where she looks at a particular transcriptional cofactor that I work on with Kristen also, the Aya family member of transcriptional cofactors and their role in the pre-metastatic niche. And in the second half of the talk, I'm going to talk more about novel regulators of metastatic clearance, all related to NK cells. So all ways that the triple negative breast cancer cells can find to evade the NK cell responses. Okay, so the major theme then of the talk today is what is allowing tumor cells to escape this NK-mediated immune surveillance in early metastasis, particularly. Okay, so one of the proteins that we work with quite a bit in the lab is a developmental regulator called the Eyes absent proteins. Like Kristen alluded to the fact that we work with 6-1. So Eyes absent are actually transcriptional cofactors that bind to the 6th family member of transcriptional proteins and activate transcription. Their names, you can guess what they're named for. Eyes absent. So in flies, when you knock out these genes, they lose their eyes. And actually in mammals, they're important for the development of numerous different organs. And if you ectopically express them along with 6 factors, like on a fly leg, you can get ectopic eye formation. In mammals, they're important for all different kidney development, muscle development, all sorts of different tissue types. And during normal development, they've been implicated in things like the epithelial-mesenchymal transition, both in the muscle and the kidney, and they've also been implicated in these processes in cancer. And they're mostly downregulated in adult tissues. So one of the reasons we got so interested in these developmental regulators is that in tumor progression, there are a lot of parallels to what happens in early embryogenesis. And a lot of these developmental regulators get reactivated in tumors and contribute to the more aggressive phenotypes of the tumors, really by reinstating developmental properties. And so this particular, the Aya proteins are actually upregulated and amplified in breast cancers. And these genes are just a number of different breast cancer data sets. And they're particularly highly expressed in triple negative breast cancer, and specifically a particular family member called Aya3. There are 4 Aya family members. Is very highly expressed in triple negative breast cancer. Okay. So I'm going to tell you a little bit about the Aya proteins. They're very complex proteins. So I told you they're transcriptional cofactors. And through their C-terminal domain, they interact with the 6th family of homeodomain transcription factors. So that's how they get to the DNA to activate transcription. But they also have intrinsic phosphatase activities. And they're dual phosphatases, but they act as phosphatases on 2 different ends of the molecule. So the C-terminal domain contains an intrinsic tyrosine phosphatase domain, but the N-terminal domain contains an associated serine threonine phosphatase activity. And they're, again, completely separable. And this also is where the transcriptional activation domain is. So they're known to have a lot of functions you can imagine because of these proteins that have multiple different effects. They have effects in the cytoplasm where they can affect motility and polarity. I'm going to talk about innate immunity. But they also have effects in the nucleus where they're known to affect DNA repair in response to DNA damage. Specifically through their tyrosine phosphatase activity, so by dephosphorylating key molecules like RAD51 and H2AX in response to DNA damage. But they're also affect cell proliferation, regulation of the transcription of a number of genes that, again, can make tumor cells more aggressive when they express these proteins. So a number of groups, including our own, have developed inhibitors that target the tyrosine phosphatase activity of this molecule. And I'm going to talk a little bit about why that's important, but why that might not be the only way to inhibit this because it leaves some other activities intact. Okay, and the other thing I want to point out real quickly is that this particular serine 3-amine phosphatase activity, well, whereas it was thought to be intrinsic to the molecule, it's actually not. So some years ago, we showed that that activity occurs through an interaction with PP2A, which is a very well-known phosphatase in the cells. And I'll tell you about why that matters also. Okay, so I'm going to give a little bit of history on some published work, and then I'll move into some newer work. So some years ago, I actually had a graduate student, Becky Vartuli, who knocked down IA3 in a number of different triple negative breast cancer cell lines. And she used some mouse cell lines that she could put back into immune competent hosts. And when she did that, she saw this dramatic decrease in the primary tumor growth of these tumor cells when she put it back into mice. And this is the 66CL4 line, which is triple negative. But surprisingly, she saw no differences in proliferation at all. And so we were expecting to see proliferative phenotypes because these had been previously said to be linked to AYA's activities. And so we recapitulated this in another model in a different immune competent background. And then we asked, well, what's going on? If there's no difference in proliferation, we also saw no difference in apoptosis. How are these tumors so different in size? And so what we did was we looked first at the immune cell compartments. And what we noticed immediately was that there was a large difference in cytotoxic CD8 positive T cells within the tumors that had IA3 knockdown. And we did this both by flow and immunohistochemistry and also by Vectra imaging. In all cases, we could see that there were increased CD8 positive T cells when you knock down IA3 in these tumors. Okay, so we did a lot of studies and ultimately ended up showing that the largest reason that AYAs contributed to the primary tumor growth was because they were suppressing the CD8 positive T cell cytotoxic response. And when you remove those CD8 positive T cells from the IA3 knockdown, from the tumors, the mice basically, where they had tumors from the IA3 knockdown cells, you can pretty much restore tumor growth almost completely just by removing the cytotoxic T cells. So we recapitulated this in multiple mouse models. And so we published this some years ago, and let me back up. And we also published mechanistically how this works. And again, because it's published, I'm going to run through this very fast, but it sets the stage for some of what I'm going to talk about today. And so what's been known for many years is that the MYC transcription factor, which I'm sure many of you know is a key oncogene in lots of different tumor types, is degraded by through this enzyme PP2A B56 alpha, dephosphorylating this S62 residue, which actually allows MYC to be recognized by the ubiquitin proteasome machinery and degraded. What we showed is that in interaction with IA3, the PP2A subunit the PP2A holoenzyme binds, instead of the B56 subunit, it binds the B55 alpha subunit. So it's just a different subunit in this holoenzyme. And that subunit changes, okay, anyway, that subunit changes the specificity of the enzyme. And instead of dephosphorylating S62, it dephosphorolates the PT58. And what that does is instead of causing MYC to be degraded, it actually leads it to be stabilized. And that stabilization of MYC was resulting in increased PD-L1 levels on the surface of the tumor cells. And that was inhibiting the CD8 positive T cell response. So in this context, AYA was affecting the primary tumor growth through affecting the adaptive immune system. And again, we published this and all the mechanistic work in 2 different papers in 2018. Okay, so this was all with respect to primary tumor growth, and my lab is actually much more interested in metastasis. And we really wanted to study metastasis when we did these experiments in the first place, but there were such large differences in the primary tumor growth that we couldn't just ignore that. So I had an MSTP student who went back into the lab and said, well, does it still play a role in metastasis in this context? So what he did is he created these knockdown lines again using the same 66CL4 model. And when he knocked it down again, we saw what we did before, massive effects on primary tumor growth. He then removed the primary tumors and looked to see what happened to metastasis. And what you can see is that there was a dramatic difference in metastasis with IA3 knockdown, but this was kind of expected. If the primary tumors are this much smaller, of course, you're going to have a difference in metastasis is what we assumed. So we didn't feel like this was really telling us if it was specifically involved in the metastatic process. So he repeated the experiment, but this time he grew all the tumors to the same size and he removed them when they were the same size. And then he asked what happens to metastasis. And when he did that, you can see while the effect is not quite as dramatic, it's still pretty dramatic. There's a pretty big effect on metastasis, even if you let the tumors get to the same size. So that made us think, yes, there's something going on in metastasis. And we wanted to understand, is that the same thing that's happening in the primary tumor, or is it something different? So the first thing he did was just RNA-seq on the cells with IA3 knockdown. And then we looked at the pathways that were associated with the knockdown. And as we anticipated, we had a big decrease in MYC target genes. We expected that given the effects we had seen on MYC. But we also had the top pathways that were falling out that were all related to NF-kappa B signaling, inflammatory responses, and immune modulation. And so we got really interested in this because we thought these pathways could be very heavily linked to metastasis. And MYC has different effects in metastasis depending on the context. So he took a look at one of the main transcription factors in the NF-kappa B pathway is REL-A. And what we did is we looked at the genes that were changed when you knock down IA3, how many of them were REL-A targets. And what you can see by this heat map is that knockdown of IA3 massively affects the targets of REL-A, suggesting that I is somehow impinging on NF-kappa B signaling. And so how is it doing that? So just to give everybody a quick primer again on NF-kappa B signaling, I'm sure many of you are familiar with this. Due to signals that occur in the outside of the cell, they'll signal into the cell, where really what I want to focus on is this IKK complex. This complex, when it gets phosphorylated, ends up in phosphorylation of IK beta alpha or IKB alpha. Phosphorylation of that leads to its ubiquitination. And when it gets ubiquitinated, it frees up P65, which is REL-A, to move to the nucleus and activate transcription. So we looked at 3 different mouse cell lines. These are all immune competent models so that we could put them back into animals. And we looked to see what happens to REL-A, this main transcription factor, when you knock down IA3. And what you can hopefully appreciate here is that in all cases, while there isn't much of an effect on the REL-A that's in the cytoplasm, there's a pretty big effect on nuclear REL-A. And there's also a decrease in this phosphorylation of IKB beta alpha, suggesting that what IA is actually doing is probably affecting the translocation of REL-A into the nucleus, less than it's affecting the transcription of these molecules. And part of the other reason we thought that is you might also notice in all of these models, IA is more cytoplasmic than it is nuclear. So again, remember, it has both roles. We know it can play a role as a transcriptional cofactor, but in this case, we suspected that the role is probably due to a phosphatase activity and not due to the transcriptional activity. So what we did is we already knew from our work with MYC that IA was interacting with B55 alpha subunit of the PP2A holoenzyme. So that's the serine threonine phosphatase. And so Annika Gustafsson, another MSTP student that actually Kristen and I co-mentor in the lab, who really works on not on this at all, but did some work to look at whether if she knocked down B55 alpha, would we see the same effects on REL-A? And sure enough, we can see decreased levels of REL-A and decreased transcription from an NF-kappa B responsive reporter when you knock down B55 alpha. Again, suggesting that it's possible that IA is working through its interaction with B55 alpha and therefore through its serine threonine phosphatase activity to affect this NF-kappa B signaling pathway. Okay, all right, so what we wanted to do then was see what's important for metastasis. Is it really the NF-kappa B pathway that's critical for the metastatic phenotypes that we're seeing? And so to do that, what we did is we restored into these cells a constitutively active IKK2 or IKK beta enzyme. So essentially, you can see this is where we've put it back. Oh, sorry, that's IA3. What you can see is the knockdown of IA3. Here's where we put it back. It's flag tagged IKK SE. And what you can appreciate, I hope, is that by adding back this constitutively active IKK, we actually restore NF-kappa B activity in these cells with IA3 knockdown. And that's also shown here. We did RNA-seq after we restored NF-kappa B signaling just to see if we could rescue it. And then we did an experiment where we put these cells back into mice orthotopically. We removed the primary tumors, and then we asked what happens over a certain time. And we asked what happens to metastasis when we restore NF-kappa B signaling downstream of IA loss. And we did this in an immune competent, actually, we also did it in an immune compromised setting, but I'm only showing you the immune competent setting because what happened was, number one, well, part of it was expected and part of it was very unexpected. So again, when we knocked down IA3, we had massive effects on primary tumor growth. But when we restored NF-kappa B signaling, we barely restored primary tumor growth. So it was doing nothing for the primary tumor. And so we actually thought at this point, this experiment is never going to work because we're not going to have Mets. But surprisingly, we massively restored metastasis. So we didn't restore it fully. This is like, these went off the charts fast, but we restored it much more than we expected to, given that the tumors were so much smaller. And so that suggested to us that NF-kappa B signaling downstream of IA3 is really important for metastasis. And of course, that affected the survival of the animals too. When we restored NF-kappa B signaling, the survival was much poorer than when we removed IA3 and had no NF-kappa B signaling. Okay, so this is where my postdoc comes into the picture and started making me think about NK cells. So Shira Rosenbaum is the postdoc in the lab who led a lot of these studies. And so what she wanted to know was, since we knew we were having effects on NF-kappa B signaling, since we knew they were so important for metastasis, she reasoned that maybe something was happening at the primary tumor very early that was affecting the metastatic site prior to the tumor cells getting there and allowing the tumor cells to be more successful once they got to the metastatic site. So she did an experiment where she took the IA3 knockdown cells. She let the tumors grow only to about 50 millimeters cubed, so very small. She then, at that time, sacked the animals, harvested the lungs, made sure she could not detect any tumor cells there using PCR-based methods, and then did flow cytometry of all the immune cells in what we're calling the pre-metastatic niche to see if anything was changed prior to the tumor cells getting there. And so what she found was really interesting. So she found a whole bunch of cells at the secondary site change, inflammatory monocytes, macrophages, dendritic cells, and cytotoxic NK cells also. And in general, though not completely, the presence of IA3 in the primary tumor was causing suppression of an anti-tumor response at the secondary site prior to us being able to detect any cells at the secondary site. So she wanted to ask, well, how is this happening? And we assumed, again, since NF-kappa B signaling downstream of IA was so important for the metastatic effect, that we would be able to find perhaps secreted molecules that were lost with IA3 knockdown, but restored with NF-kappa B signaling that might play a role. And so she looked back at she did some cytokine activity arrays, and then she picked a subset of cytokines here that, again, lost with IA3 knockdown and restored with NF-kappa B signaling restoration. And in particular, she focused in on this cytokine CCL2. And there's a lot here. You really only have to look at a couple of the samples. So what she did, this is looking at by ELISA CCL2 levels. Here's CCL2 levels in our wild type cells, control cells. Here's our IA3 knockdown. And then this is with restoration of NF-kappa B signaling. She could restore CCL2 in the media. So she then wanted to ask, and this happened in numerous different contexts. We looked at both human triple negative breast cancer contexts, but also other mouse systems too. And so what she wanted to ask was an experiment that I actually told her not to do because I thought this was never going to work, but she did it anyway. It's good. She didn't listen to me. And what she did was she wanted to know whether she could just add back conditioned media to the mice without any tumor cells ever there, and see whether she could change how the microenvironment looked at the secondary site without tumors being injected. And so she took conditioned media, either control media where she had added back CCL2, IA3 knockdown media, or media where IA3 was knocked down and she added back CCL2. And then she collected the media and she filtered it. And she injected it for 7 days into mice. Then she harvested their lungs and did flow cytometry. And when she did this, she got some pretty cool results. So I first will say the reason she honed in on NK cells was because this experiment did not cause the changes in any of the other immune microenvironment changes like macrophages or monocytes that we had seen with the primary tumor present. It didn't cause those changes, but it did affect the NK cells. And so when she isolated the NK cells, she also looked at markers of a cytotoxic NK cells, so their activity, so TNF alpha and interferon. And what you can see is that when she put back the media where she knocked down IA3, those were dramatically increased, suggesting you get more cytotoxic NK cells when you lose IA3. And when she added back CCL2 into those cells and took that conditioned media, she didn't totally restore it back to the background levels, but she restored it quite a bit. And if she looked at the levels of cytotoxic NK cells by markers, by flow in the lungs, again, knockdown of IA3 increased the levels of cytotoxic NK cells, and adding back the conditioned media that had the CCL2 in the context of the knockdowns, restore that back to more normal levels. So all of this suggested that there is something being secreted in the media by IA3, and that something is CCL2, and that's educating and changing the function of the NK cells at the secondary site, which is a bit unusual because normally you think of CCL2 as affecting macrophages. And I mean, we can talk about this afterwards. There's things I don't quite understand about this. But anyway, I'm thinking you're going to ask me something about that. But anyway, okay. So the other problem we had, though, you might have noticed on those graphs that there aren't a lot of NK cells in the lungs. It's not like it's a huge number, right? And so we were concerned, and reviewers were concerned, because actually this was just published, that there weren't enough NK cells to make a difference. And so we had to show that even small numbers of NK cells in the lungs are going to make a difference to metastatic outgrowth. And so what Shira did was then took mice in which she put in the conditioned media either from the scrambled or the IA3 knockdown cells. This is a bit of a hard experiment to explain. She then injected the media 7 days prior to putting cells in. And then a few days prior to putting the cells in, she depleted NK cells in groups of these mice so that we could look at whether NK cells were important for the response. And then she injected into the tail vein of these mice cells that were wild type. So this is really important because we figured we couldn't inject the knockdown cells into the mice because they probably wouldn't grow at all. And so we needed something to grow at the secondary site. So it's a bit of a complicated experiment. They're getting media from cells that are knocked down as well as wild type, but they're getting wild type cells. So they're still producing a little bit of the CCL2, but they haven't been conditioned with that media. And then she continued the depletion of NK cells, and she sacked the animals at a humane endpoint and looked at what happened both to metastasis and a number of different parameters, like looking at the NK cells. But I'm going to show you the metastasis results. So what you can see here is that when you inject with the media where you knock down where IA3 is knocked down, you do get a decrease. It was not significant, but it's trending that way. And I'm not, again, surprised about this because remember, they got wild type cells. But when you depleted the NK cells, you restored metastasis, suggesting even those small number of NK cells matter. So the problem in this first experiment that we did is that we didn't actually deplete the NK cells in the control condition. And so you don't know if they're contributing just in general. We didn't think they were because there were so few NK cells in the lungs, but cytotoxic NK cells, but we did the experiment again. And in this case, we depleted the NK cells in the animals that got the scrambled media. And you see that made no difference. Again, when you put media in with IA3 knockdown, you got less mets, but when you added back, when you depleted NK cells in the context of having the media from the knockdown cells, then you get more metastasis again. So that tells you that even a very small number of NK cells at the secondary site makes a difference for these cells being able to grow out and cause metastasis. Okay, so is this relevant at all to human breast cancer? Obviously, we're doing this all in mouse models because they need to be immune competent. And so what we did is took a Metabrick data set and looked at all the nearest neighbor genes to IA3 and then ran them through pathway analysis to see, are we seeing the same pathways activated in human breast cancer that we can see in the mouse models? And sure enough, you can see that MYC targets come out right on top, and so does NF-kappa B signaling, suggesting that in the context of these tumors, IAs, at least they associate with in the human tumors, these two key pro-tumorigenic and pro-metastatic pathways. Okay, so we just published this a couple months ago in Science Advances. And so if you want more details, you can see it there. And so I'm just going to summarize something quickly and then move into some newer data. So what we've shown for IA3's role in tumor progression is, again, that it affects both NF-kappa B signaling and MYC signaling in the primary tumor, that signaling not only affects the immune microenvironment in the primary tumor, but also leads to secretion of molecules that affect the immune microenvironment at the secondary site. And that leads to more outgrowth at the secondary site and more metastasis. But this brought up a lot of additional questions for us. So first of all, both mechanisms, both the MYC mechanism and the NF-kappa B mechanism, appear to act through an interaction of IA with B55 alpha and PP2A. So with this phosphatase that is often considered a tumor suppressive phosphatase, but we would argue that when it's interacting with IA, some of its activities become tumor promotional. And this makes this very complex in terms of thinking how to drug or target. And that's what we've been thinking about. Can we find a way to inhibit this process to suppress both primary tumor growth and metastasis? And so I'm going to tell you a couple ways we've been trying to do this. And so one of the ways, the first ways we've done it, I told you at the very beginning that our lab and others have developed inhibitors against the tyrosine phosphatase activity of IA. So many years ago, MYC was first actually shown to be controlled by 6 and IAs, so I am talking about 6-1 a little, transcriptionally in kidney development. So these molecules were first identified for their mechanisms developmentally, right? And that was published many, many years ago. Also, it was shown that the IA phosphatase activity stabilizes MYC also during kidney development, not quite as long ago, but a while ago. And again, we showed that IA3 is acting on MYC to increase its stability via this interaction with PP2A. So that's all working through the serine threonine phosphatase, which is totally separable from the tyrosine phosphatase. But in both of these contexts, the tyrosine phosphatase has been implicated in these processes. So people have shown that the tyrosine phosphatase activity is critical for the ability of IAs to activate transcription of MYC. And other groups have shown that the tyrosine phosphatase activity, including our own, impinges on the serine threonine phosphatase activity of IAs. And so the first thing we reasoned is, well, if we inhibit the tyrosine phosphatase activity, we might be able to take out the other activities, which would hopefully help to remove some of these tumor promotional processes. And so quite a few years ago, in collaboration with the Zhao lab, that's also in the Department of Biochemistry in Colorado, we did a high throughput screen to look for inhibitors of the tyrosine phosphatase activity of IA. And we identified this class. You can see, even if you don't know anything about chemistry, because I don't, but I could see all these molecules are super related. And those were the only molecules that came out of the screen. And I actually thought this was awesome when it happened, because I thought, we've nailed it. This has to be real. It's only this. What I've discovered since that time is that it is not good at all if you only have one chemical backbone like to work off of. So anyway, not as good as I thought, but I was excited at the time. And so we identified this molecule, and through a lot of work done over the years, we've actually shown exactly how it works. So Ray's lab, in collaboration with A-Star in Singapore, we've solved the crystal structure of the molecule bound to IA2. And by the way, I should mention, when we did the screen, we did it against IA2, not IA3, which is the IA that I showed you is important in all these breast cancer models. And that was for various reasons, because we were using IA2 in other models. Anyway, it's an allosteric inhibitor. So what it actually does is it binds to the back of IA, and it pushes into a pocket that affects the catalytic domain. And the catalytic domain needs to bind magnesium to act, and it can't bind magnesium when this molecule is bound. So because of that, it doesn't act as a tyrosine phosphatase. So it's an allosteric inhibitor, which gives it high specificity, which is great, because phosphatase inhibitors are usually super hard to develop because the active sites are shallow, and oftentimes the small molecules will just like kill every phosphatase, and that's not a good thing. So we were first excited about that until we started to test it against the other IAs. And then it turned out to be so specific that it only bound IA2 and not any of the other IA family members. And we actually had wanted to hit the other IA family members because there was an indication that IA3, which happens to be relevant in the models that I tested, is not the only IA that's relevant in breast or other cancers. And we figured out exactly how that works. And the reason is it binds from the structure. We knew that it bound specifically to this phenylalanine that sits in this pocket behind the active site in IA2, and that all the other IAs have a tyrosine there instead of a phenylalanine. And if you change that phenylalanine to a tyrosine in IA2, our molecule doesn't work against IA2 anymore. So super specific, which is great and not so great at the same time. So it won't target IA3 in breast cancer. So I couldn't use this to test whether it was going to make a difference for any of the things I just showed you. So this is going to be a quick change in direction just for a second. But it's kind of the lesson for the trainees that you just have to go where the data takes you, because then we had an inhibitor that wasn't going to work in our models. So we had to figure out what to do. And so it turned out another colleague of ours in Colorado, who is a pediatric oncologist who focuses on brain tumors, had done at that time some microarray studies and had shown that IA2 was really highly expressed in a particular subtype of pediatric medulloblastoma called group 3, or now people refer to it more as MYC medulloblastoma. And that was interesting to us because, again, we knew that it was a MYC regulator, and now it's really high in this MYC subtype of pediatric medulloblastoma that actually is a very devastating subtype. It's a subtype that more kids die of than the other subtypes. And so I'm going to run through this quickly because we published this a year ago or so, but we basically did a number of studies to look at whether IA2 expression was is higher in MYC medulloblastoma than the other subtypes. And you can see here, this is MYC expression in this group 3. IA2 expression is very high in that same subtype. If you then look at levels in cell lines that are derived from group 3 medulloblastoma versus other subtypes, this is a sonic hedgehog subtype of medulloblastoma. Again, you see IA2 is very high in this group, as is MYC. And so we decided, all right, let's test our inhibitor in this context. This context looks to be very reliant on IA2. And so I'm just going to show you the bottom line of these experiments. What we did is we did get lucky about one thing with this. Look how we got lucky with a couple things, but this inhibitor crosses the blood-brain barrier. We did a bunch of PK analysis. So what we did was intracranial tumor experiments where we injected the tumors into the mice. And then we treated with one of our inhibitors for over time. And what you can see is we got a pretty dramatic reduction in tumor growth in these mice with the inhibitor. And what was cool is that when we took the tumors out of the mice and stained for MYC, we could see a really dramatic decrease in MYC levels, suggesting we're on target. We can't test if we're on target of IA2 as easily as we can target or test whether we're on target for one of its targets. So this was all great. But how do we target other IAs? So we've been working on this. And so we're working with a chemist who's at CU Boulder. He's actually does structure-based drug design. So Ray's lab, who's at my campus, has been doing a lot of structures of analogs. He's been taking that and trying to find analogs that he can get to interact less with that phenylalanine. And he's actually developed a set now that inhibit IA1 and IA3. We still can't hit IA4. And they don't inhibit it as well as IA2, but we're on our way with that. So we're working on that. It's not good enough to test yet in vivo. We're also trying to directly target the IA PP2A interface. And I'm going to tell you about that in a second. And we are identifying additional modulators of the immune tumor microenvironment, which I'll get to in a minute also. Okay, so let me tell you quickly about this. How are we doing this? So again, this is mostly done in collaboration with Ray Zhao's lab, and she's a biochemist and structural biologist. And so what they did first was just start with mapping the domains of IA3 that interact with B55 alpha. The idea being that maybe we could develop peptides that could disrupt that interaction. And so Chris Elderman, who was an MSTP student in our lab, mapped it really to this domain in here, which is interesting because many years ago, when it was first said that IA was a serine threonine phosphatase, it was mapped to this domain also. So we're in good shape here, I think. It corroborates that data. And then she saw the crystal structure of PP2A, B55 alpha, the holoenzyme bound to IA3. Unfortunately, the domain of IA3 that's interacting with B55 alpha is like a very unstructured domain. So she only got a little bit of that structure, but she got it. And she did that almost at the exact same time as Wolfgang Petty and Rebecca Page solved the structure of IA3 bound to PP2A. So we published sort of back to back in the last couple of months at the same time, not back to back. And so what was interesting about this is that, so this is IA3 bound to the B55 alpha subunit. And around just very recently, last year, somebody through informatic approaches developed a putative inhibitor, peptide inhibitor of B55 based on a bunch of substrates that it was binding. And so what she did was she made that peptide and then saw the structure of that peptide inhibitor bound to the enzyme and overlaid that with IA. And you can see that this peptide inhibitor, which is in pink here, is overlaying where IA binds. So we reasoned, okay, this inhibitor is going to disrupt IA3 from binding to PP2A. So she actually tested that in her lab. She used mass photometry. So here we're just measuring the molecular weight of IA3 alone, of B55 alone. You put the two together, it shifts because it gets heavier. And you add in the peptide and you remove that shift, suggesting that we're going to be able to use this peptide to inhibit the interaction. I can tell you we have not gone in vivo. We've just started this, and there's a lot to do to work out on how you get a peptide in vivo and get it to be stable. But we do think that this could be an approach to try to inhibit specifically that interaction that we could then test in all of our immune competent models. And the reason we think this is going to work is because of this. So years ago, when someone else had pointed out that H79A mattered before we had the structure and mapped it, we actually showed that that mutation in IA3 disrupted its ability to interact with PP2A. And we showed that if you took mice that had a wild type IA3, again, they get lots of Mets. If you knock down IA3, that's decreased. If you add back a wild type IA3, the Mets are restored. But if you add back this IA3 that can't interact with PP2A, now you don't restore the metastases. So again, this suggests to us that if we can disrupt this interaction, we're going to affect metastasis. Whether the B55 alpha peptide is the best way to do it is something we can talk about if we have time. Okay, so what are the challenges ahead with this? We still need to identify what the targets are of this IA3-PP2A enzyme in the NF-kappa B pathway. We aren't sure what it's dephosphorylating to affect activity of this pathway. What we do know from some pull down experiments is that if you look at this IP here, here's IA3, it appears to be in the IKK complex. So it's sitting there with the IKK subunits. And that's interesting because long ago, actually, nobody ever pursued this. In the Drosophila field, someone showed that IAs are interacting with IKKs, and nobody really went on to try to figure out what it's doing. So we think that this is relevant. We just don't know what the substrates are. We also need to better understand how IAs alter the PP2A activity. So is it just by binding to that different subunit, it takes it to different places? We have to understand whether it is specifically targetable. There are reasons that that peptide inhibitor is not going to be as specific as we would like. And the other thing is that we don't think that our tyrosine phosphatase activity is going to fully inhibit the NF-kappa B pathway activation by IA. So we've got to think about other ways to hit this. We're also looking at PROTACs and other ways to just get rid of IA. Okay, so in the last few minutes, I'm going to talk quickly about now how we sort of shifted to thinking about NK cells and an important role of NK cells at the secondary site. So when Shira did this work, she got just interested in general. Could we find ways sort of more quickly? IAs are not going to be druggable really fast. I mean, this is something we're working on, but we're far from. And so she wanted to know, are there different ways that we could figure out what in the tumor cells sort of reprogram the NK cells that we could maybe try to target or inhibit? And so again, her focus was, how do the NK cells actually contribute to metastatic clearance, and can we figure out a way to inhibit that? Okay, so what she first wanted to do was just make sure that the model system that she was testing actually behaved like she thought it would. And that is that NK cells are the first responders to the metastatic cells when they get there, but that T cells are going to have a role, a big role, right? But in later metastasis. And so she took this EO771 model. So this is a cell line that you can put into C57 black 6 mice, sorry. And she just depleted either NK cells or CD8 positive T cells, and she looked to see what happened to tumor, to the outgrowth of these metastatic cells at the secondary sites, so in the lung. And so what she was able to show is what she thought. Remember, these are early time points, so she's taken this all fast. We know that the CD8 positive T cells are going to be important longer term, right? And what you can see is that when you deplete the NK cells in the very early stages, you massively increase your metastatic outgrowth. When you deplete the T cells, it's starting to come up, right? It does affect it, but it's slower. It's the next responder as compared to the controls. And so this suggested to her that this was going to be a really good model in which she could do a screen. And so she decided to design an in vivo screen to look at whether she could identify things that allow tumor cells to escape NK cell mediated immune surveillance. All right, so she set up this CRISPR-Cas9 screen, and she used only druggable targets. So she got a library that was all druggable targets, so targets that have drugs already available that she could use. And she took this library, she put it into the cells. It had about, it was targeting about 1,024 genes, and she had a non-targeting guides and controls, et cetera. And then she injected this either into IgG-treated mice or mice in which she depleted the NK cells. And then she waited until she had some cells in the lungs. She took those cells out, and then she did sequencing in order to see what remained. And essentially, by doing the screen, any guides that were enriched in the NK cell depleted group would target genes that we should be drugging, right, to boost the NK cell response early. And so she did the screen. She got a number of hits out of the screen. And then she did a secondary screen from the primary screen because we had too many hits to know what to go after. Again, to see whether she could get some of the same hits and if she could help narrow the hits. And in this screen, she used only 118 genes. So she had a number of targeting guides. And again, just the same experiment again. in which she put in the druggable lentiviral guide library and then either depleted the mice of NK cells or had her control mice. And when she did that, she also included a number of positive controls that we know affect NK cell response. We didn't have those in the first screen, and we knew that would be important. This is an in vivo screen. So things that fall out, a lot of times there are reasons they fall out, not for the reasons you're looking for. So the nice thing was that in her hits, the top targets were all these genes that are important for NK cell mediated detection of tumor cells. So we got all those hits in the screen. It was very clear the screen worked well. And in addition, we got a number of other hits in this screen. And the hits that she's been most focused on is this hit PIT-PNA. And she's really focused on that more than these two that came up even higher, GLO1 and SARC, because these have already been pretty significantly implicated in breast cancer metastasis, whereas PIT-PNA, there's very little known about how it affects metastasis and really nothing known about how it might affect the immune response. And so what is PIT-PNA? It's the protein is called PIT-P alpha. It's a protein that transfers phosphatidylinositols from ER to various organelles and lysosomes. You can tell this postdoc is making me go in all sorts of directions that I know nothing about. But anyway, it's been fun. So it's really important in signaling in the cells. It regulates membrane trafficking, receptor signaling, autophagy, transcription, mRNA transport, cytoskeletal dynamics, all sorts of things, right? And so she reasoned that this was a really interesting protein to study because it's also really important for secretion of molecules and exosomes and things that might allow crosstalk between tumor cells and NK cells. And that's what she's trying to look for. Okay, and there are, again, inhibitors of this molecule that exist. Okay, so this is really new work. So I'm just going to tell you the little bit she's gotten into with this so far. But what she did do before she pursued it too much is also just look to see whether it correlated at all with activated NK cells. And it does inversely correlate, not terribly strongly, but strongly enough to make her go after it. And it's also linked to poorer survival in lung metastases or lung metastases-free survival, which is, again, what she was interested in looking at. So she decided she was going to pursue this to see whether it could be a new molecule that we could target to basically enhance the NK cell response to metastatic cells. And so what she did is knock down PIT-P alpha in breast cancer cells, same model that she used to do the screen. And this just shows you her knockdown. And then she injected these into the mice and then did IVUS imaging to look at METs. And she also did flow cytometry to look at NK cells, T cells, a whole bunch of immune cell types to see if it was affecting them. And when she looked at the NK cells, she looked at a number of markers of NK cells that really tell you about NK cell maturation from sort of an immature NK cell to a very cytotoxic NK cell. And so when you knock down PIT-PNA in TNBC cells and do this experiment, what you find is that there is a change in markers that suggests that the NK cells are maturing to become more cytotoxic. So in fact, it looks like expression of PIT-PNA in the tumor cells is affecting the activity and the maturation of the NK cells. She also had an increase in markers on the NK cells that suggest that they're more cytotoxic, markers like CD107. So you can see again, when her knockdown cells were injected, the NK cells had more of the CD107. But I should say, and I won't go into this too much because I want to end soon, but she did see effects on other immune cells too. So it wasn't like the effects were only on the NK cells. So for example, CD4 positive T cells were somewhat increased when you knock down PIT-P and alpha in the tumor cells. And this is something she still needs to understand better. And so what happens to metastasis? Again, we're looking only at early metastasis. We know that long-term, this is not going to be enough. So ultimately, when we think about therapies, if we want to target NK cells, will make NK cells more cytotoxic, I think it would have to be combined with CD8 positive T cell, immune checkpoint therapies. And together, those might be very strong in the context of this kind of cancer. And so when she looks at early metastasis again, when she knocks down PIT-PNA, she sees a decrease in early metastasis. Okay, and that's about as far as she is. So we don't understand how it works mechanistically. She's trying to look at that. We need to do experiments where we try to use the drugs that inhibit PIT-PNA alone and in conjunction with immune checkpoint therapies. And all these things are things that are underway that she's doing. So with that, let's see, what else do we, just to summarize, again, I've given you some data about IA3 in the pre-metastatic niche. and now some more newer data from a screen that my postdoc has done to look at new molecules or other molecules that are druggable that might inhibit the NK ability to clear metastatic cells. And we have a lot to understand. I just talked about this a little bit. How does PIT-P alpha influence NK cells? You know, what are the effects only in the metastases, or do they do something in the primary tumor? We have some data on this already. It does look like it affects the primary tumor. Investigating crosstalk between NK cells and other immune cell types. And then again, I talked about therapeutic targeting. Okay, so with that, I want to thank everyone in the lab and all my collaborators who've done all this work. Again, Connor Hughes started some of the early work on NF-kappa B. Shira came in and really did everything to do with NK cells, pre-metastatic niche and metastatic niche. The Zhao lab, which has been absolutely critical for biochemical and structural work, Wang lab for medicinal chemistry, and then all my other collaborators. We have an advocate who works pretty closely with us now that we've moved more into this immune effects. And then, of course, all my funding. And with that, I'll take any questions.
I have microphones for anyone who has a question. Please raise your hand.
Thanks for a fantastic talk. I have two questions. The first one is, because you haven't gone in enough directions, maybe we can add one more.
Oh, no.
Have you thought about the role of EVs, exosomes, etc., in creating the niche?
Yes, and we absolutely have to do that. David Leiden was at our institution a couple weeks ago, actually, and he was like, "Why are you not looking at that?"
Yeah.
Okay, well, I will. I promise.
Yeah.
Yes, I mean, we should get there. We just have never done anything in that space, so it was easier for us first to think about secreted molecules, but I think it's probably really important.
Yeah.
So we're about to embark on that.
Cool. The other thing is, I don't know if you're probably aware, another group at CSU has looked at treatment of metastasis in the context of bone tumors. And so they did a clinical trial on dogs that had advanced metastatic disease with losartan and a split kinase inhibitor, which targets CCL2 and probably other things. And they saw some really nice signals.
Yeah, was that Mike Bernaris? Do you know who that was?
No, I don't think Mike was involved in that. This is Steve Dow and
Oh,
Dan Regan.
yeah. I have heard about it a little bit, and I should talk to them about that.
It's interesting because their concept is that it's targeting CCL2 secreted by the macrophage compartment.
Right.
But I wonder if there's any effect,
on
that's
NK
comparable
cells
on NK cells, or maybe on CCL2 secretion by the tumor cells rather than exclusively. So
that's actually a really good point. I should circle back to them. I'm always hearing about their dog studies, which is so cool, right? And I don't feel like we do. I mean, our cancer center interacts with them, obviously, but I feel like we could do a lot more between the two campuses.
Any other questions, Scott?
So I was fascinated by that experiment with the injection of the conditioned medium IP and then seeing the effects on the NK cells in the lung. Were other tissues evaluated in that experiment? Was this a lung specific effect?
She did. Okay, so in the media experiment, I can't remember if she looked at the liver also. In the original experiment, she did. And the major effects were in the lungs and not in the liver or other tissues, which is a little bit, I don't quite understand that, right? Because if this is secreted, you would expect the effects. And so, but I'm not sure she did it with the conditioned media experiment, and we should do that. She probably has all those tissues saved because I think if it's a systemic effect like this, there have to be other tissues that are affected. And so I think we need to look at that more carefully. So it's a good question.
All right, we've got a question from our virtual audience, Dr. Stanger. Great talk. I was wondering if you think CCL2 inhibits NK cells in the PMN across tumor types, or is this something specific to triple negative breast cancer?
I don't know the answer to that question. It's a good question, and I'd like to look at that because also we were a bit surprised that this was the effect we were seeing on CCL2. So in the literature, there are different effects of CCL2 on NK. I mean, mostly, again, people have looked at it with macrophages, and on NK cells has been shown to be either inhibitory or stimulatory. So I don't know the answer. I haven't tested that, so I can't answer. I would like to see if it holds up in more models and more tumor types. There's a question here. Go ahead, Doug.
That was great. So I want to ask a question about something you brought up in the beginning and right at the end. So as of today, the only approved immunotherapy for triple negative breast cancer is PD-1 inhibitor, pembrolizumab. So very early on, you described an association between IA3 and PD-L1 expression. PD-L1 expression is required for pembrolizumab to be prescribed. So have you looked at either thing? PD-L1 expression and IA3 expression amplification in triple negative breast cancer. And have you ever used a PD-L1 inhibitor in your model systems?
We have not, and I don't know why, because this is the one thing I keep saying, like, we got to do this. We will do this very soon. I think it's really important to do, right? I will say that I think the initial paper where we showed that it affects PD-L1, and that it does that in part through MYC, that's PD-L1 on the tumor cells only and not in the microenvironment. And I don't know, I have to say, I don't think that's the entire explanation for what's going on with the CD8 positive T cells in the tumors that express IA3. But nonetheless, I absolutely think we have to try those experiments. I don't know why we haven't yet. I think people have just been going in other directions, but we will try it. I'll tell you because now, especially with these newer inhibitors in that screen, I think there's a number of contexts in which we need to try those inhibitors in conjunction with either IA3, IA3 knockdowns, or wild type where we try to inhibit IA3 with the tyrosine phosphatase, or with some of these newer ones like PNA, which we have a drug for too. We need to try these experiments. So I'll tell you when it's obviously something we need to do.
Yeah, this was a really good talk. I have a question about the pediatric medulloblastoma, the molecule that you use for the EVA2. You said it was, with your results, brain penetrant. Did you look at the leptomeninges? Did you test that to see if the metastasis, that it affects metastasis? And
we did not. No.
What you see is just the brain.
We're just looking at the primary tumor in the brain by luciferase imaging, right? And those studies are so fast, like within 2 weeks, or it's so much faster than the breast cancer studies. We have to sack the mice because it's in the brain, that we haven't spent a lot of time digging down into that. And that's another thing. I collaborate with another group to do the intracranial transplants, and because this isn't something my lab has done. And so I think we need to start doing more careful studies like that, but we just haven't done that yet.
And the other question, just a technical question, on that CRISPR screen that you did, is that something you put together, or is that something, because you said it was a druggable thing? So I'm assuming that everything in there has at least one or two compounds you can get your hands on. So
our institution has a functional genomics shared resource, and the functional genomics core put together this library for somebody and went through and picked a bunch of molecules that are druggable and made it for us. So we have it at the institution.
So it's was it picked for just this, or I guess is a library a generic
It's generic. So they had made it generic. They had made it for another group that wanted to do a druggable CRISPR in vivo screen.
Right.
And then we knew they had it and said, hey, we want to use that library.
Thanks.
All right, great. Any other questions? Oh.
Really awesome work. Thank you. So quick question, and sort of following up on something that I think a couple other people have asked. It looked like most of the metastasis effects that you saw were in the lungs. Did you really look anywhere else, or was that impeded by the models and the time points that you have to use?
Again, early on, we did look in other sites, but then later we stopped because most of the effects were in the lung. And I think that's model specific. I don't think it's because there couldn't be these effects in other sites. I think the model just goes more to the lungs. But again, getting back to that other question, certainly if there's something systemic, it shouldn't only be affecting the lungs. So I'd like to go back and start looking at that more specifically now that we have the model set, but we haven't really explored it deeply yet.
Cool, thank you.
All right, we're out of time, so let's say thank you one more time to Dr. Ford.
Thank you.